The cancer hypothesis. The landscape of early Earth was barren, consisting of rock and water. About 2.7 billion years ago, our ancestors, ancient eukaryotes, lived in water, fermenting sugar as energy. Rich oxygen accumulated in the atmosphere about 500 million years ago, and ancient organisms evolved from anaerobic into aerobic metabolism. Many new species evolved after massive oxygenation of the Earth. An aerobic metabolism is 19 times more efficient than an anaerobic metabolism. Viruses and mammals evolved with aerobic metabolisms. Many viruses invaded mammals and left their genetic fragments inside their host's genome. The viral fragments are in an inactive state and not much impacts on cell function, only providing limited functions. Example, development of the placenta when required. In cell division, if the daughter cell's DNA is not perfectly replicated, the viral genetic fragment could be active. Then a new cell becomes a cancer cell, with the traits of that virus it arose from. After a cancer cell developed, other viral gene fragments could randomly activate and develop another new cancer cell. This is the reason why cancer genomes are not the same for two cancer patients. As the cancer progresses further, the ancient genes become active and create new cancer with ancient eukaryotes genes. The ancient gene cancers have stronger innate immunity in comparison to viral gene cancers. Therefore, ancient gene cancers will prevail over most viral gene cancers. The late-stage cancers are dominated by ancient gene cancer. Here is a comparison of late-stage cancer and ancient eukaryote. 1. Metabolize glucose by fermentation. Late-stage cancer metabolize glucose by fermentation, similar to ancient eukaryote. 2. No cells adhesion. Normal cells have stickiness between the cells to keep them together. Ancient eukaryotes lived in water, therefore have no stickiness trait. Ancient gene cancer has no adhesion and easily spreads to other organs. 3. Proliferates on soft tissues. If a cancer cell is placed on soft matrix, the tumour proliferates, similar to ancient eukaryote in water. If the cancer cell moved onto stiff matrix, tumour is dormant, similar to eukaryote washed onto a rock. If the cancer cell moved back to a soft matrix, the tumour proliferates again, similar to a eukaryote washed back into the water. This is the reason why most cancers spread into soft tissue, such as the lung or breast. 4. Dormant under stressful environment. Organisms' dormancy is one of the best self-preservation in evolution history. The ancient genes cancer went dormant under stressful environmental conditions. Dormancy minimizes the DNA damage treatments. Any remaining surviving tumour will regrow again after the treatment is over. 5. Proliferation is a natural trait of a unicellular organism. Proliferation under a suitable environment is a natural trait of ancient eukaryotes. Ancient gene cancers have same proliferating traits as ancient eukaryotes. Cancer hypothesis summary. The ancient eukaryote genes are deeply embedded in mammals' genome. The viral genetic relics are embedded in mammalian junk genes. Although viruses could induce cancer, mostly the viral genetic relics are initiated and driven cancer development. As cancers progress further, the ancient genes incorporate into newer cancer cells. The ancient gene cancers will prevail over most viral gene cancers. The late-stage cancers have the same traits as ancient eukaryotes. Cancer is a genetically modified organism, GMO, by undesirable dormant genes in genome. Einstein said the simplest explanation is usually the correct one. Reference: Chan JCY, Hypothesis of Cancer Weakening and Origin. Journal of Cancer, March 2015.